Welcome to this explainer. Look, we so often think of our social lives entirely in psychological terms, right? Our feelings, our thoughts, the messy nuances of our relationships. But beneath all that conscious experience is this vast, hidden network of biological machinery. Today, we're doing a rigorous deep dive into the neural architecture of social learning. We're going to explore the actual, physical, hardwired mechanics that dictate exactly how our brains learn from, bond with, and truly understand one another. If you're ready to unpack the cellular how and why of human connection, you're in the right place. Let's get into it. So, I want you to start by asking yourself this. How does your brain actually learn from others? Think about the instant, automatic nature of it. You see someone touch a hot stove and you flinch before you even have time to process why. Or you see a friend give this genuine smile and bam, you immediately feel a spark of warmth yourself. What is physically happening inside your head to make that possible? See, there's this massive gap between our conscious experience of empathy and the subconscious, structural processes firing off in the brain. Social learning isn't just some soft, abstract psychological concept. It's a deeply biological, structural reality. Okay, let's dive into our roadmap for today. First, mirror neurons and empathy. Second, decoding theory of mind. Third, the chemistry of bonding. Fourth, socialization and learned fear. And finally, number 5, the power of imprinting. Module 1, Mirror Neurons and Empathy. Let's start with the mirror neuron. This is a highly specialized cell located in the brain's premotor area, or the PMA. Now, to grasp why discovering this completely revolutionized neuroscience back in the 90s, we have to look at how we used to view the brain. For the longest time, motor areas like the PMA were thought to be strictly outbound, like a one-way street, just sending commands to your muscles to move. But the discovery of mirror neurons, total game changer. It showed us that these motor areas are actually processing the visual input of other individuals doing things. They actively bridge the gap between perception and action, basically linking the actions we observe directly into our brain's own motor programs. To picture this, imagine a macaque monkey grasping a peanut. A specific neuron fires in its premotor area to execute that exact hand movement. Makes sense, right? But here's the wild part. If that same monkey sits completely still and just watches another monkey grasp a peanut, that exact same neuron fires again. It's this brilliant dual activation. The brain is reacting as if it's performing the act itself. And it's not just passively mimicking the geometry of the movement. These neurons are highly goal-oriented. They fire when mapping the intent to grasp the food. If the monkey just observes aimless waving without a target, the neuron stays completely quiet. It's mapping the purpose, not just the motion. This brings us to what's known as the simulation hypothesis. To put it in tech terms, we are essentially using our own motor circuits to run internal software programs that interpret the actions of others. Let's look at a microexample. If you watch someone reach for a glass of water, you don't have to consciously sit there and deduce, hand is moving to glass, therefore they must be thirsty. No way. Your brain instantly runs a neural simulation of that exact action in your own premotor area. By completely bypassing conscious thought, this simulation lets you instantly decode their goals and intentions. This right here, it's the biological bedrock of empathy. Module 2, Decoding Theory of Mind. So social learning eventually has to move way beyond physical, tangible actions. It requires a much higher level social ability that we call theory of mind. And that is the capacity to interpret completely unobservable mental states. We're talking hidden desires, complex intentions, and underlying beliefs. Predicting what someone might be thinking, that requires a massive leap in brain processing power. From an evolutionary perspective, this incredible ability correlates directly with the huge expansion of the frontal and temporal association areas in the primate brain. It's like we effectively added the biological servers needed to process all this invisible social data. This evolution of mental state interpretation relies heavily on the prefrontal cortex acting as this massive centralized data hub. It pulls in and integrates incredibly diverse sensory information so we can interpret nuanced behavior. And we know exactly how structural this is by looking at what happens when the hardware breaks. When a physical lesion or damage occurs in these prefrontal areas, individuals can literally lose their social tact. They can no longer read a room or pick up on the unstated intentions of others. A biological injury can completely rewrite an individual's personality, essentially evaporating their social self, simply because the physical hardware required to maintain it has been broken. Module 3, The Chemistry of Bonding. let's shift gears and look at the neuropeptides oxytocin and vasopressin, drawing on some incredible insights from studies on monogamous voles. Now, these pituitary hormones are absolutely critical for attachment, but what's truly fascinating here is where they actually operate. In monogamous species, the receptors for these chemicals are densely concentrated in the striatum and the nucleus accumbens. Under normal circumstances, these brain reward centers fire to reinforce essential survival behaviors, like eating a high-calorie meal or drinking water when you're super parched. But oxytocin and vasopressin, they effectively hijack these survival pathways. Biology wires us so that social connection literally feels like a biological necessity for survival. Just look at the structural differences in a marmoset brain before and after fatherhood. Once a marmoset becomes a father, there's this significant, easily measurable increase in the dendritic spine density inside his prefrontal cortex. Now, dendritic spines are essentially the physical branches that connect neurons together. More spines mean a literal physical increase in wiring and communication pathways. The massive social responsibility of fatherhood doesn't just change the monkey's daily habits, it demands entirely new hardware. The brain is physically growing new neural architecture to meet those heavy social demands.
Module 4. Socialization and Learned Fear.
You know, a massive portion of social learning is simply acquiring knowledge about what to avoid without getting hurt yourself. Imagine being an early human. If you had to actually get bitten by a venomous snake to learn that it was deadly, well, you wouldn't survive very long, would you? Instead, you observe the danger or the consequences of that danger in someone else. Immediately, your amygdala, which is basically the brain's threat detection center, forms a powerful emotional memory of that danger. You successfully avoid the threat without ever having to pay the physical cost. Through socialization, you literally download another individual's survival data directly into your own amygdala. It's incredibly efficient. But the amygdala manages way more than just predators. In primate societies, the most persistent threats are often other members of the group. Navigating a highly complex social hierarchy requires a perfectly calibrated level of aggression and threat response. If a dominant monkey suffers a lesion to the amygdala, they lose that aggressive drive. They become placid. Unable to mount the necessary threat response to challenges, they rapidly drop straight to the bottom of the social hierarchy. It perfectly illustrates how our social standing in the animal kingdom is governed by very specific neural centers. Finally, Module 5, the power of imprinting. In many species, the most rapid and permanent form of social learning happens through imprinting. But there's a catch. It is strictly bound to a critical period. This is a highly sensitive, finite developmental window. Early sensory experiences, like the very first moving image a young animal sees, become permanently etched into the nervous system. And biological timelines, they are entirely unforgiving. The window opens, the brain records the environment to form a lifelong attachment, and then the window slams shut. If that window closes before imprinting occurs, the capacity for that specific type of attachment is permanently lost. Let's unpack this concept of phenotypic permanence. A phenotype is the observable trait or behavior of an organism, and permanence means the environment has reached right into the biology and permanently set the dial. Those early sensory experiences don't just form a learned habit that you can easily unlearn later in life. They represent a permanent structural modification of the nervous system. Our earliest environmental interactions literally hardwire the neural circuits to ensure survival, essentially setting the concrete foundation for exactly who we become and how we attach to others for the rest of our lives.
So we've explored the premotor area, mapping intent, the prefrontal cortex, deciphering hidden minds, the amygdala, downloading fear, and the intense chemical reward systems that compel us to connect. Biology shapes absolutely every facet of how we interact. So I want to leave you with a final thought to digest today. If our earliest environments permanently hardwire our neural circuits, setting the strict biological parameters for exactly how we learn, bond, and fear, how much of our social behavior is truly our own choice? I challenge you to ponder those boundaries between biological programming and free will as you navigate your own social world today.